And one of the issues that it's mainly that FDA was asking is, so what is a meaningful itch reduction? And I think this is a very valid question because you want to develop a drug that reduces itch. And a lot of trials show, oh, the p-value is significant, but what is it meaning that the NRS from a 0 to 10, it went down by 1? Even if it is statistically significant, it doesn't mean anything to the patient. And this is a study that I worked with Alexa Kimball, with Eli Lilly, and just published this year, looking at the numeric rating scale for looking to validate it versus the DLQI, the quality of life measures in dermatology, for 4 clinical trials they had with their ixekizumab. So it's the largest study so far on patients with psoriasis. And what we found is that a 4-point change demonstrated a level of clinically meaningful improvement in itch severity. And why is it so important? Because this will be most probably the benchmark that FDA was asking for a meaningful antipruritic drug. And I can tell you that in other conditions, the same in atopic eczema or any other drug trials, most probably, I think that the FDA would kind of adopt this benchmark because it sounds that this is a very significant and meaningful parameter for our patients. So I think that now I need to go into a bit the basic science of it as to why we need to understand mechanisms of chronic itch, because that will enable us to effectively treat itch. So we need to examine the transmission of itch from skin to brain, and there are actually 3 components. Remember that most of the itch emanates from the upper layers of the skin. So today when you talked about panniculitis, sarcoid panniculitis would never itch because it's too deep. But epidermal conditions or cellulitis would not be itchy, but erysipelas or impetigo would be itchy because the process is very superficial on the upper layers of the skin. So remember that keratinocytes are involved in it, immune cells, not just T-cell lymphocytes, but we recently have a lot of evidence that neutrophils have a role in itch, as well as the neural system. Clearly, everything is transferred by the nerves, and there are 2 types of nerves that transmit itch. One, you are well familiar, is the histamine pathway, but it's actually more relevant to acute types of itch. The more interesting in terms of chronic itch is the non-histaminergic pathway. This is activated by spicules of a plant called cowhage that activate proteases. A part 2 receptor is on the nerves. The neural transmission goes through the lateral spinothalamic tract. It goes to the spinal nerves, to the dorsal horn, and from there it decussates. For you guys who are residents, you remember from neuroscience, I know it's a bit difficult for the folks that have been dermatologists for so many years to understand this pathway, but it's a very similar pathway to pain. And one of the questions that I was always concerned about was what happens in the brain. And I was initially, when I started to ask for funding, dermatologists were saying, why is this guy measuring brain activity in itch? Because it's an immune disease, kind of. But it's clear that the neural system is the common pathway that transmits itch. And that's why the issue that interests me was that there are a lot of components of the itch. So there is a sensory component of itch transmission, but there is also some emotional aspects. You know, when you see your patients when they're stressed, they have more itch, and there's also issues of scratching and the pleasurability of itching a area. So an itchy area, scratching an itchy area. And one of the first studies we've done, and this was funded by NIH, was understanding the 2 major pathways of itch, the non-histaminergic pathway using these spicules of a plant cowhage, and histaminergic. So there are good news and bad news. The good news is that we know how this pathway is activated in the brain, and that's extremely important. The bad news is that there's no just one center. There are numerous areas that are activated, so it's not going to be one drug that would cover everything. But I think that it's important to understand that there are areas in the brain that are involved in sensation, the somatosensory cortex. There are areas in the brain that are involved in unpleasant sensation, like the anterior cingulate cortex, as well as areas that are involved in activity of what we call premotor areas that are the supplementary motor cortex. Now, if you would be in a pain brain imaging session, you would think that the brain imaging results are very similar to the ones that are in itch. However, there are some unique features. One of the areas called the precuneus, let's see if that works. This precuneus is part of the posterior cingulate cortex. In pain, it's actually deactivated, and itch almost always is activated. Another important aspect is histamine activates really limited areas in comparison to the cowhage, which is more important because this is the activation in most types of chronic itch. Now, as I mentioned, you can't divorce itch from scratch, and there is a component of pleasurability of scratching an itch. So it has a powerful effect that cannot be ignored. The itch has a powerful effect also on motivation. It kind of causes us to continue scratching. I mean, when you have an insect bite, or when residents start seeing a scabies patient, they start scratching themselves, and I did that too. So what are the central mechanisms of the reward pleasure generated from scratching an itch? And this is another study that we've looked in. First, we looked at passive scratching. That's not the real life. How many people ask someone else to scratch them? So one of my fellow at the time was saying, Gil, we should do active scratching. We should ask the patients or the subjects that we induce itch and look at how they scratch themselves to better understand the mechanism. So when we did the passive scratching way back more than 10 years ago, we've shown that really scratching inhibits some of the areas that I showed you that were activated, such as the anterior cingulate cortex. And, but doing the active scratching, we're able to see a lot more information and also correlate it to the effect on itch intensities. And what was striking is that there's a lot of elements of activation of areas in the brain that are involved in reward, in addictive behavior. So in a way, scratching is rewarding because it activates a lot of these areas in the brain, including the substantia nigra and the red nucleus, as well as the striatum. And recently, we looked at patients, these were healthy before, asking patients with chronic itch when we induce itch, and these mainly were atopic, psoriasis, and uremic patients, and brain image their activity of scratching versus healthy subject. And we found again that the reward of a patient with chronic itch scratching is a lot more higher than in healthy subjects, and the brain shows that too. So the areas in the striatum, what you see in the yellow here, is areas of the striatum, and this is the premotor, the supplementary motor cortex in yellow. These were highly more active in patients with chronic itch versus healthy subjects. So we think that there are differences in brain activation between these areas, and it's important to then better understand why this chronic itch and scratching is a vicious circle. And the biggest mistake you could say to your patients is stop scratching. They can't stop that. It's like telling a drug addict to stop using drugs without really approaching it with a more comprehensive treatment. Another issue is a very common phenomena in patients with chronic itch. The lightest touch of the skin can evoke or exacerbate an ongoing itch. Like a lot of times the patients tell you that we put clothing on ourselves and we start itching. This is actually a neurosensitivity called Alloknesis, and it's defined as itch and scratching behavior evoked by a stimuli that normally does not do an itch. And it's very common among atopic eczema patients, but any other types of chronic itch. And this is an example of one of my patients who has this phenomena. He has the butterfly sign, so you can see that there's sparing of the areas where he cannot scratch, and in the pattern of a butterfly. And the issue with this patient, and I have a lot of patients like that, is they state that the worst part of the itch is not the spontaneous itch, it's the skin sensitivity that they suffer. So the question is, how can the nervous system get this itch so wrong? So I kind of give it as a comparison to if you imagine the nervous system in the body is hardwired like your house. So if you have an itch caused, for example, for dry skin or eczema, the nerves that transmit itch are firing, and they transfer this information to the junction box. This wiring system is overcharged, and in these patients, it's like the nerves are acting wacky in terms of that. But clearly, the situation is more kind of complex than what I describe here. But it's as if someone, came into your home and shut the off button in the electricity, and it's just continuously firing. And the nerves in this type of approach condition, they start morphing and changing, and it's what we call neuroplasticity. This is very common in chronic pain patients. So this is not something that we invent in terms of itch process, but it's very common in pain. And what we did also is looking in patients' brains of atopic eczema with chronic itch and imaged the itch induction and looked at it versus healthy subjects. And in fact, we've seen that there are differences also in these areas, not just in the peripheral nerves and in the spinal cord, but also in the brain. Some of these areas, such as the anterior cingulate cortex and the dorsolateral prefrontal cortex, are involved in this neuroplasticity. So it's not surprising that the activity in these areas in patients is much higher. And this will explain to you later on some of the treatment regimens that I have. Now, there are a lot of new receptors involved in itch. In this area, almost every week now, we have some new information about receptors. It's unbelievable how this field is exploding. But two concepts that I would like to convey to you, because it will be easier for you to understand some of the treatments we do, is that there are ion channels of two groups that are transmitting itch. And Scott knows that I like good food, but these are channels that are related to food too. So one is the chili pepper receptor, that's the TRPV1, that's the capsaicin receptor. That's related more to histaminergic itch. But more interesting is the TRPA1, it's the wasabi receptor. This is a receptor that is an ion channel that is highly associated with atopic eczema. So again, these could be targets for treatments of peripherally to reduce itch.